Okay, in this video, we are going to be looking at the cross-bridge cycle. Uh, that background noise is my dog chewing her bone, so sorry about that. Uh, but anyhow, in a previous note page, you would have read about the sliding filament theory of muscle contraction or the sliding filament model of muscle contraction. And in this model, uh, we see that muscle contraction is the result of the thick filament sliding the thin filament towards the M line of the sarcomere. And as the thin filaments slide, the sarcomere grows shorter. And we see that because we know that because the Z discs are drawn closer to each other, the H zone disappears, and that's some of the evidence that suggests that the thin filaments are being slid. So the cross-bridge cycle actually is the mechanism by which the thick filament does slide the thin filament during contraction. So let's take a look at it. Our textbook looks at it as being comprised of 4 different steps. Step 1, Step 2, Step 3, Step 4. And keep in mind this is a cycle, so once we finish Step 4, we come back to Step 1. Another implication of it being a cycle is that we could really start at any of these steps and it would still make sense. It does make a little bit more sense if we start here. So before this can happen, actin's myosin binding site has to be exposed. So normally when the muscle is relaxed, these active sites are blocked by tropomyosin, which is the orangish goldish thread here that we see. Tropomyosin is attached to troponin, those little yellow spheres, and in order to uncover actin's myosin binding site, what needs to happen is the sarcoplasmic reticula need to release calcium within the cell. That calcium is going to bind to troponin. When it binds to troponin, troponin is going to shift, and that pulls tropomyosin out of the way of actin's myosin binding site. So that has to happen before this cross-bridge cycle can occur. So we've imagined that that has happened, so let's get into the cross-bridge cycle. The first step our textbook calls cross-bridge formation. And what's going on here is that the energized myosin heads, and they're energized because ATP has been hydrolyzed, they reach across and attach to actin. So remember, there's myosin binding sites on the actin molecule, and then there's actin binding sites on the myosin molecule. So the myosin head reaches across and binds to actin. That's cross-bridge formation. That refers to the fact that there is now a physical connection between the thick filament and the thin filament, the cross-bridge. All right. So then the next step is this myosin head still has energy left over from hydrolyzing that molecule of ATP. And what it's going to do with that remaining energy is it's now going to pivot. So we can see this arrow indicating that the myosin head has pivoted. Remember, there's a hinge region here, and the hinge region allows the head to pivot. And that's what we see happening here. Well, the pivoting is what's sliding the thin filament one increment towards the M line of the sarcomere. Now we're looking at what's going on at the right-hand side of the sarcomere. So we see the arrow sliding to the left here. The M line would be somewhere over here. Keep in mind that this is also happening on the left side of the sarcomere, but in the opposite direction. So thin filaments are being slid from both directions. We're only seeing one direction here. Well, whenever the myosin head pivots, it then releases ADP and phosphate from that ATP binding site. And that's actually an important step here. That will allow a new ATP to come along and bind to the myosin head in the third step, which is called cross-bridge detachment. So with cross-bridge detachment, we see that the myosin head has been released from actin. And what causes it to release is this new molecule of ATP has come along and bound to the ATP binding site on myosin. Okay, so the myosin head has released. A new ATP is bound. What do we do with that new ATP? Well, we move on to step 4, which they're calling cocking of the myosin head. And the new ATP is hydrolyzed. That now re-energizes the myosin head for a next round of cross-bridge cycle activity. So now this myosin head is energized once again. We're going to go back to step 1. It's going to reach out and bind to actin's myosin binding site, and we have a cross-bridge again. This is going to go on repeatedly during a muscle contraction. It's kind of like mind-boggling how many times over and over again this happens just in a simple muscle contraction.